Okay, everybody, last installment of this chapter. So the last few things I want to talk about, and there's just a few more slides, have to do with continuation of our discussion on violations of Hardy-Weinberg assumptions. So in the last episode, so to speak, we were talking about inbreeding and how it is a problem for populations. And we can actually calculate something. It's kind of complex to calculate, so we're not going to calculate it. But population geneticists calculate a statistic called F. And this F is the probability that two alleles are identical by descent. And here's what we mean by that. Let's take this example here where you have these two individuals that mated here and they've had progeny. And as you can see, the result of inbreeding, if we look at this allele here and we trace it through this pedigree to here, and then again through this pedigree to here, we can see that we have a homozygous, and they got really each copy of this allele from a common ancestor. So these alleles are common by descent, okay, or identical by descent. These alleles are identical by state. So in this case, you have this A1 allele that came through and ultimately ended up here. And here you have outbreeding and an A1 allele here. So it's still an A1 allele, it still codes for the same thing, but it was inherited not by a common ancestor, but through a process of outbreeding. And so if we can figure out the probability that alleles are identical by descent, that gives us some measure of whether inbreeding is occurring. And that also tells us something about the health of that population. So inbreeding coefficients, or F is what the statistic is, they can range between 0 and 1. 0 means there's no inbreeding whatsoever going on in that population. The alleles in that population do not arise from any intermingling of familial folks, okay? 1 means complete inbreeding, all right? And population geneticists are really interested in this, particularly if you're managing, say, a fish stock, or if you're managing, say, an endangered pack of wolves. You would be very interested in the statistic as a measurement of how inbred your population is, because that tells you something about the health of that population. So for example, cheetahs have a very, very inbred population. I don't think their inbreeding coefficient is 1, but it's darn close because they were a very small population. They started inbreeding with one another. And if a disease comes along or something happens, cheetahs are going to be in a lot of trouble because they're very genetically similar to one another due to inbreeding. So this is just a chart that shows you, I remember how I said that inbreeding increases homozygotes, and we saw that in the little video, particularly recessive homozygotes, which therefore, if it's increasing homozygotes, then it must be decreasing heterozygotes. So often, population geneticists are interested in measuring the heterozygosity of a population to get a handle on how, what the genetic variation of a population is and how healthy a population is. So this just shows you how much you reduce heterozygosity depending on what kind of inbreeding you have going on. So for example, if you have sort of in plants where they're self-fertilizing all the time, or maybe an asexual reproduction, or maybe a sponge that self-fertilizes or something like that, you very quickly, because there's a lot of inbreeding, you very quickly diminish the proportion of heterozygotes. And self-fertilization actually has an inbreeding coefficient of 1, okay? Siblings, you can see that it's a little bit slower. Their inbreeding coefficient is 0.25, first cousins, et cetera, et cetera. Again, this calculation's a little involved, but please understand that if you have an inbreeding coefficient of 1, that is a completely inbred population, versus an inbreeding coefficient of 0 is no inbreeding whatsoever. And again, this is important because it measures the genetic variation of a population, and it tells us something about the genetic health of a population, all right? Let's talk about a different assumption this time. Let's talk about assumption 2, which is migration, or we're gonna call it gene flow, movement of genes between populations. Now, I told you that Hardy-Weinberg assumes that there is no migration between populations because, for example, if we go back to our original diagram I had for you way up here, okay? And I showed you that these numbers here, let's go back this way. Come on, little mouse, there we go. These numbers here are different than what we're expecting, right? But maybe they're different because we had a whole bunch of students transfer in and they brought with them different alleles, right? They brought with them a different proportion of alleles from, maybe they all transferred in from Winthrop, and there's a higher proportion of people in Winthrop that have this genotype, and so that's what's affecting our outcome. So migration can cause problems with Hardy-Weinberg equilibrium. And the amount of genetic change due to migration depends on the difference in the allelic frequencies and the extent of migration. So if we look, for example, in population 1 here and population 2 here, and we can see that the little a allele is present in 1, 2, 3, 4, 5 out of the whole. And we're going to assume that that's true here, 1, 2, 3, 4, 5. Okay, so these are actually at very similar proportions, but so if we add a sample from this population to our population, it might not really change our allele frequencies very much. But what if we had a population over here where instead of having only 5 like little a alleles, it had 15 or something like that? And then some of these individuals moved over. You could imagine that that would change our P and our Q. It would change our frequencies, wouldn't it? So migration can cause populations to change over time. And of course, population change over time is what evolution is. So there are 2 possible effects that we can get from migration. One thing that can happen is it can cause gene pools to become more and more similar. If we start having a lot of migration between these 2 populations, over time they're just going to mix up and they're going to reach equilibrium. They'll start functioning like a single population and they'll mix up over time. So these populations will become more and more similar. In the short term, it might change things, but in the long term, they're going to get all mixed up together. The other thing that migration can do is it can increase genetic diversity. Okay, so let's say that you had maybe just a few of these little a alleles, but you had some immigrants from another population come in and bring in more of those little a alleles. Or maybe you had some immigrants from another population come in and they brought a whole new 3rd allele that you didn't even think about. So migration can also cause evolution. It can cause deviations from our Hardy-Weinberg equilibrium. Now, this one is one of the assumptions we make, and that would maybe be violated by a small university like us. And that is, we assume when we're talking about Hardy-Weinberg equilibrium that we are drawing from a population that is quite large in size. Now, why is that important? Well, if you have a small population, you're vulnerable to something called genetic drift. And genetic drift is really well described by this image right here. So in this scenario, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So it's a population of 20, okay? And we have 10 of each allele, 10 of the red allele and 10 of the blue allele. But we have just a really small number of individuals in total, don't we? So if we sample some of these individuals, because the population is so small, it would be very easy to accidentally oversample one versus the other, which is what we've done here, right? If we oversample and then we let reproduction happen, you can see that suddenly our proportions have shifted, haven't they? After one round of reproduction and mating, maybe more individuals with the blue alleles mated with one another, and now we've shifted our allele frequencies due to something called sampling error. Sampling error is more likely in small populations when you take small samples. Okay, and you can see that, okay, well, let's see, we'll let mating go on again here, and oh, maybe sampling error has occurred again here, and it's occurred again here. Over time, you see what's happened? We've drifted, the gene frequency or the allele frequency has drifted away from where we had, looks like we have a P of about 0.5 and a Q of 0.5. So half and half, to where we have a P of 1. There's no Q at all, is there? Okay. When you only have one allele possible because of genetic drift and you've ended up, you don't even have that second allele anymore, we say that this allele is fixed. This is called fixation. So this allele is fixed in the population. Now, if you move on from here and you have more reproduction, you're sure enough going to end up with all blues henceforward because there's no reds to draw from. The only thing that would introduce some variation at this point would be a mutation, right? So that's why we say it's important to assume that we have a large population size, because large population sizes are less vulnerable to sampling error. They're less vulnerable to that right there, okay? Now, in thinking about genetic drift, there's a couple of special cases of genetic drift that may actually make this a little bit more obvious to you guys. So we see genetic drift a lot with two phenomena, one called the founder effect and one called the bottleneck effect. So I'm going to start with the founder effect. So in the let's look here. Let's say we have a mother population where we have P's and Q's. Let's say that the P's are yellow, okay? And the Q's are purple. So that's down here. Now let's say maybe these are birds or maybe these are dragonflies. And so a small subset of them move, and maybe they're going to populate a new area. Maybe it's an island, or maybe it's far away over a mountain. And so this new population is not going to have any more interaction with the mother population. There will be no migration, no gene flow between these two populations. These are now isolated. Well, the sample here that was taken, because this was a small population to begin with, and this sample that was taken, this sample isn't representative of this population, is it? It doesn't look like the proportions of purple to yellows are the same. And so as we move forward, we're going to see that the proportions, we're going to end up with a lot more P than we have Q, aren't we? The allelic frequencies are going to shift because of this founder effect, all right? A genetic bottleneck is very similar, only you usually have some kind of catastrophe that comes along and knocks out part of the population. So I mentioned like cheetahs this happens with, but this is a great example. So we've got some green and some tan bugs, and some jerk comes along and steps on some of them. And the resulting final population, because this was a small population to begin with, it wasn't well mixed. So this jerk's foot disproportionately affected the greens, and now we don't have the same proportion of alleles in the population here. So founder effect and bottleneck effect, this would be bottleneck, this would be founder, okay? And you can see why those names are descriptive. Those are both examples of how genetic drift might occur. In your homework, I've given you a great animation and some questions to answer following that animation to help you understand this concept a little bit further, okay? The next assumption I want to talk about is mutation, and I'm not going to say a whole lot about mutation, but it makes sense that let's say that we have allele big A and we have allele little a. You can imagine that a mutation can come along and it might create a third allele, maybe A prime, right? Because we end up with a whole nother allele. Another thing that can happen is, let's say that you have a situation where they call the first allele G1 and the second allele G2. Sometimes you can have mutations from G1 into G2. Maybe that's a common mutation. Maybe it's a missense mutation. And that might affect the frequencies of your alleles. Now, the only thing I want to say about this is that mutation tends to have a very small effect on Hardy-Weinberg equilibrium. And that is because mutation rates tend to be very, very low. Okay, so while mutation is an important source of genetic variation, it takes many, many, many generations for a mutation to become prevalent enough in a population that it becomes an allele that would be part of our Hardy-Weinberg calculations as population geneticists, okay? The very last assumption or violation of an assumption that I want to mention is natural selection. Something that can alter our allele frequencies is, of course, natural selection. And this is a great example. So here we have some mice. They've moved from wherever they were living. They're now living on this very barren, gray hillside. And in our mouse population, there are tan mice and there are gray mice. Maybe where they came from, this was fine, but now the tan mice stand out. And these birds are more able to see the tan mice, so they start picking them off. That means that the gray mice are left behind at a greater rate, and they're the ones that reproduce, okay? So natural selection is acting on this population to diminish these alleles and I guess enhance or increase these alleles. I won't say enhance, I'll say increase those alleles. So these are going down each generation, these are going up each generation. This, if natural selection is present, then it violates Hardy-Weinberg equilibrium as well. Okay, so that's what I want you to know about population genetics. Congratulations, you've made it to the end of the course. So a few announcements. All three of these videos, of course, are going to be posted, and hopefully you've gotten this far so you know that. The PowerPoint is posted. The homework is posted. I've given you a couple of extra questions on this homework, like I did on the last one. They can only help you and not hurt you, okay? So everyone, you know, hopefully will get a little bit of bonus out of this. So if there's, you know, 30 possible points, but I've given you 32, that means everybody's going to basically get 2 points for completing the homework. All right. So that was what I did last time as well. I don't know if you guys picked up on that. Everything is due to me at the end of the day on April 28th. That is reading day. Then the last thing I'll point out is I am putting together a study guide for you guys. I'm working hard to get that all together for you. I will probably have it for you over the weekend or Monday morning at the very latest. Good luck. Let me know if you need any help with anything. Don't forget to turn in your papers. Many of you already have, and I will talk to you soon. Bye-bye.